Today let's discuss about the sodium potassium pump. It is a biochemical pump that transports the sodium and potassium ions across the cell membrane against the concentration gradients. So it is seen in the animal cells, the membrane of the animal cells. There are two types of transports, passive transport as well as active transport. Passive transport occurs spontaneously without any expense of energy. Example is diffusion through a semi-permeable membrane. For example, in this case, there are two type of molecules. The first type of molecules and it is partitioned with something, and if the partition is removed, we can see that there is spontaneous diffusion from a high concentration region to the low concentration region. Here this violet colored molecules are at a high concentration in this compartment, and it can diffuse easily to the other compartment because there it is having a low concentration. Similarly, these yellow molecules can migrate to the other portion, that is from a high concentration to the low concentration. So it is a natural or spontaneous property or a spontaneous process to migrate from a high concentration to the low concentration. Such kind of transport is known as active transport. It does not require any energy for such transport. But if we have to shift from a low concentration to a high concentration, it requires energy. Such kind of transport is known as active transport. That is, when a solute is moved against the concentration or electrochemical gradient, it will consume energy, metabolic energy, and it is termed as active transport. The sodium potassium pump is such kind of pump which is an active transport mechanism. Here the sodium from a low concentration region will be transported to a high concentration region, and potassium from the extracellular fluid where it is in a low concentration, it will be transported to the interior of the cell where it is in a high concentration. So it is against the concentration gradient that the sodium and potassium ions are getting transported with the help of this sodium potassium pump. So it's a biochemical pump, a protein that hydrolyzes ATP. With the help of ATP, this pump is working in order to transport a particular solute through a membrane to generate an electrical gradient. It is actually discovered in 1957 by Danish scientist Jens Christian Skov, who was awarded Nobel Prize in Chemistry for his work in 1997. This is a person, he got Nobel Prize after 40 years after the discovery of this pump. It helped to understand how the ions get into and out of the cells, especially in the case of excitable cells like nerve cells, which depend on this pump to respond to stimuli and transport impulses. So the main function of this biochemical pump is to move sodium ions out of the cell and potassium ions into the cell against their concentration gradient. So if we describe the mechanism, the first step is the 3 sodium ions, 3 Na plus ions will be attached to the cavities or will get inserted to the opening of the sodium potassium pump, which was towards the inner portion of the cell, and then the ATP molecules will get attached to it. It will undergo a hydrolysis, and this phosphorus linkage will cause some conformational change in the protein ATPase, sodium potassium ATPase, and the inner opening will be closed, and there will be an opening towards outwards. So this will help in the release of Na plus ion to the extracellular fluids, and then the potassium ions, 2 K plus ions, will enter into the cell, into the sodium potassium pump, and this will cause a dephosphorylation. That means this phosphate group will be eliminated, and again it will cause some conformational change, and the opening will be again converted or back to the inner portion, and this will lead to the potassium ions to get released into the cell, inside the cell. Again this will be repeated. The same process will be repeated again and again. So if we want to check the mechanism stepwise, first the pump binds with 3 intracellular Na plus ions and ATP. Then ATP will be hydrolyzed, leading to the phosphorylation of the pump. Then conformational change in the pump exposes the Na plus ions to the outside, and the phosphorylated form of the pump has low affinity for Na plus ions, so they will be released to the extracellular fluids. Then the pump binds with 2 K plus ions. This causes dephosphorylation of the pump and reverting it to its previous conformational state, and the transportation of K plus ions into the cell. The unphosphorylated form of the pump has high affinity for Na plus, so the K plus ions will be released to the inside the cell. Then again the ATP binds and the process will repeat in a cyclic manner until the gradient is reached. Then about the function of the sodium potassium pump, it helps to maintain the concentration of sodium ions and potassium ions across the cell and the electrochemical gradient. The failure of this pump will result in swelling of the cell. It controls the neuron activity produced as a result of electrochemical gradient produced across the cell membrane. Again, the kidney have high level of expression of sodium potassium ATPase, and this sodium gradient is necessary for the kidney to filter the waste products in blood, reabsorb amino acids, reabsorb glucose, regulate electrolyte levels in the blood, and in order to maintain pH. So these are the functions of sodium potassium pump. Thank you.